guess I'm
Bernie
next.
Sherman.
I thank the organizers for giving me the opportunity to talk about the kinds of questions that my lab is interested in dealing with. And this actually comes from a long-time collaboration I've had with Ray Guillory, who unfortunately died last year. This collaboration has lasted several decades, and we've concentrated on trying to come up with new ideas about thalamocortical relationships. We published a number of papers and a few books on this. My lab has attempted, there we go. My lab has attempted to address a number of these questions. I don't have time to go into very much detail. I will give a few examples of experiments aimed at testing some of these questions. But most of the talk is going to be directed at identifying what we think are interesting questions and some speculations, and not really a lot of data. I also want to indicate that much of what we're doing now involves a collaboration with a colleague of mine at Chicago, Jason McLean. Okay, so for the outline of what I want to talk about, I'll start off with basic thalamic circuitry. I'll then explain how we've divided thalamic nuclei into two types we call first and higher order. And this has components for cortical processing, which I will get to. And then I'm going to switch gears fairly dramatically and consider what the role of the driving inputs to thalamus. And by driving inputs to thalamus, we mean the inputs that carry the information to be relayed to cortex. For instance, the retinol input to the lateral geniculate nucleus, and how they might be related to efferent's copies. And I want to mention now that this is going to be very speculative, and my goal anyway...
You just want to turn the microphone.
Oh, okay. Can you hear me now? Yeah. Okay, good. Yeah, my goal here is not necessarily to get you to agree with me. Frankly, that doesn't matter to me so much. Is to make sure that, or to try to get across ideas that we think need to be addressed that have been pretty much ignored up till now. And I'll end with some of these speculations. Okay, so this is a cartoon view of the human thalamus. This would be the back to the front of the left thalamus. These shown in color here represent the relay nuclei. These contain cells that project to cortex. Each projects to one or a small number of cortical areas. The thalamic reticular nucleus actually would sit flush against the lateral surface. It's pulled away and cut away, so you can see the relay nuclei. This nucleus contains GABAergic inhibitory neurons that don't project to cortex. Instead, they innervate relay cells and provide powerful inhibition there. 2 points I want to emphasize is that as far as we know, all cortical areas receive the thalamic input, and virtually all information reaching cortex must pass through thalamus. There are some exceptions to this, but this means that the thalamus sits as a very strategic location for information flow, and if information doesn't get through thalamus, cortex won't receive it, you won't be consciously aware of it, you won't perceive it, and we think this is the thalamus plays a very important role in attention. I'm going to draw attention to two thalamic nuclei that are involved in visual relays. The lateral geniculate nucleus obviously relays retinal information, and this is a monkey brain that with a computer assist has been flattened out. This is V1, that gets the input from the lateral geniculate nucleus. The pulvinar, which is the largest relay in the thalamus, innervates all of the extrastriate visual areas, nearly the back half of cortex. So we think of this as a visual relay also, and the size of the pulvinar is about 30 to 50 times larger than the LGN by volume, and I'll get back to the size difference later. So using the LGN as an exemplar of thalamic circuitry, here we have the relay from retina to relay cell up to middle layers of cortex. This is layer 4, 5, and 6. These axons use glutamate as their neurotransmitter. We have local GABAergic inputs from the thalamic reticular nucleus and local interneurons that are GABAergic. We have a feedback from layer 6 that's also glutamatergic. Finally, we have inputs from the brainstem reticular formation that bring classic modulatory inputs to thalamus, cholinergic, noradrenergic, and so on. And I want to emphasize that these relay cells get two glutamatergic inputs, but they're quite different functionally. Theron Briggs, after lunch, will talk a lot more about this feedback, but we think of the retinal input as the driving input, and this is providing a modulatory function, much like cholinergic input to these relay cells. The difference being that the classic modulators are very diffusely organized, but glutamatergic modulation provides the only pathways that have a high degree of topography to give you modulation, which is needed for such things as spatial attention. Other thalamic nuclei are organized pretty much the same as far as the non-retinal inputs. The main difference is the input to be relayed. There are differences here to be sure, but I'm going to ignore them. To a first approximation, it's pretty much the same. So if we can identify the input to be relayed, we can learn a lot about a thalamic nucleus and its function. And that has been a bit of a challenge. Now I'm going to use the idea of different sources of driver input to divide thalamus into first and higher order components. So here's our LGN again, and here is the Pulvinar. And the Pulvinar has, again, the same non-retinal inputs pretty much, with minor differences. But the question is, what is the driving input? And just like the retinal input to LGN, which terminates with very large synapses on proximal dendrites, often in complex synaptic zones known as glomeruli, we've known for decades that there is a similar kind of synapse on Pulvinar cells. The question has been, what is the source of these synapses? And work over the last decade or so has made it pretty clear that for most, if not all, Pulvinar cells, the driving input comes from layer 5 of cortex itself. So we have an interesting scheme here where all thalamic relays get a feedback from layer 6 that is a modulatory input, but Pulvinar cells, in addition to that cortical input, get another one from layer 5 that's not organized as a feedback, but in this example is a feed forward. And these axons have the same property as the retinal input to LGN. So the suggestion then is that Pulvinar provides a relay from one cortical area to another. We call LGN an exemplar as a first order relay because it's a first relay of a particular kind of information, a cortex, visual in this case. And Pulvinar we call a higher order relay because it's relaying information not from a subcortical source, but from cortex itself between cortical areas. And I should point out that we think the Pulvinar relays information not just between V1 and V2, but you can go up the hierarchy. Remember how much larger Pulvinar is than LGN, so there's plenty of bandwidth for this. We see this division not just in the visual system, but also the somatosensory system, where the equivalent of the LGN, the first order relay, is VPM and VPL. The posterior medial group is the higher order relay. In the auditory system, the ventral division of the medial geniculate is first order, the dorsal division is higher order, and so on. But one point I want to emphasize is that while we have these transthalamic pathways to connect visual cortical areas, we also have direct connections. We don't have very many examples, about 5 or so, mostly from the mouse. But for those examples, anytime there's a direct connection between cortical areas, there's a parallel transthalamic one. And this raises a number of questions. First, how often is this parallel organization seen? Or to rephrase the question, how often are areas connected just by the direct or the transthalamic pathway? Yeah.
So I have a vague memory of reading that it's a very specific set of layer 5 cells.
Yes,
that
layer 5B.
Right? Okay.
And the other layer 5 cells don't project subcortically, they project other cortical areas.
And Mary, the same neuron that gets input, say from V1, doesn't go back to V1?
Is there a projection up back to V1?
Not from Pulvinar.
Yeah, that's an open question. And one question that we don't have an answer to is whether this is feed forward, there may be feedback pathways involving this also. That's an open question we're trying to resolve. So one question again is how common this parallel organization is. The second question is what's different in the nature of the information between the direct and transthalamic pathways. And the third question is why bother to send one information route through thalamus? We don't have answers to those questions, some partial suggestions, but I think those are important questions to keep in mind. Now I'm going to just show some evidence for these transthalamic pathways. First, a slice experiment that's been published some time ago. These are brain slices in the mouse. They're about 0.5 millimeter thick, and they're cut at a very precise angle, in this case in the somatosensory system, to preserve this circuit between the primary somatosensory cortex, the higher order thalamic relay, and the secondary area. We can't do this in the visual system for topological reasons. And I want to emphasize that because of the angle of the cut, we've artifactually cut the direct pathway, so you won't see evidence for any connection here. But that's an artifact of the slice. What we're doing is uncaging glutamate over layer 5, and when you do that, you see some activity in thalamus, but also activity in the second somatosensory area, and it can be followed to the third area. And I will say, since this has all been published, I'm not going to show you, that if the uncaging is done in layer 6 or above layer 5, you see no activation of the higher cortical areas. To show that this depends on the thalamic relay, we can put a drop of DNQX, which is an AMPA blocker, blocking the synapse, on the slice, and when you do that, you completely obliterate the responses in the higher cortical area. And when you wash the DNQX away, you restore most of the activation in the higher areas. So what this tells us is that this pathway is a very robust pathway, a very strong one, and certainly this is consistent with the idea that this pathway is carrying important information. I will tell you again, since this has all been published, if we cut the slice at a different angle that preserves the direct pathway, you can see evidence for a strong connection there as well. So we do have parallel organization in these circuits. Now I'm going to switch gears to talk about the evidence for this in awake behaving animals. We use a Cree-line mouse where Cree is provided in layer 5 cells, which allows you to specifically target these layer 5 cells for particular agents. And the one we're interested in, these layer 5 cells, of course, give rise to the cortico-thalamocortical pathways. We're using a DREAD approach here. Designer receptors exclusively activated by designer drugs. You can target these designer receptors to these layer 5 cells specifically. These are modified muscarinic receptors. When you put inject the viral vector into, in this case, V1, you get the layer 5 cells taking up these receptors, and they're sent through the entire cell, including to their terminals, wherever they are. The agonist for these receptors is CNO, and when CNO is placed on these receptors or on the cell body, you open potassium channels leading to a strong hyperpolarization, and this will, at synaptic terminals, will greatly reduce transmitter release. So that's the idea behind these experiments. To show how this works in a slice, in this case in the somatosensory system, we put the DREAD receptors and channel rhodopsin, both Cree-dependent, into S1. So the channel rhodopsin and the DREAD receptors, the channel rhodopsin is labeled with the green tag, the DREAD receptors with the red tag, and you can see that it's layer 5 cells that are taking these up. You can see their terminals, the channel rhodopsin labeled terminals, and the DREAD terminals in the higher order relay. You don't see them in the first order relay because layer 5 doesn't project there. When we activate the synapse between the layer 5 and a cell we patched in POM with channel rhodopsin, you see the large inward currents with each pulse of light. When we add CNO to the slice, we get a tremendous reduction in the synapse as expected, about 90%. And we can repeat this experiment in the visual system, which is the system we'll use in vivo. Same kind of experiment, same kind of reduction in the layer 5.
Where are you recording here?
I'm sorry?
Where are you recording?
Here we're recording in POM. We patch a cell that's in this labeled area. Here we're doing it in the Pulvinar. Okay, so the preparation we're using here is a mouse that has the layer 5 Cree-line, and that is crossed to a GCamp mouse so that we have a calcium signal in cortical neurons. We put a window over V2, so we're able to monitor the responses of hundreds of cortical cells simultaneously. The animal's head fixed on a treadmill, so he's awake and behaving, and although we haven't done behavior yet, one of the plans we hope to fulfill is to get some more sophisticated behavior to test some of our ideas. The animal is looking at a visual stimulus, in this case a drifting grating, and this is an example of what you see. These cells in V2, these are V2 cells, are very selective for direction and orientation of movement. I'm just showing 4 directions here. We normally do 8 to 12. And you can plot in these examples a high degree of direction and orientation selectivity. So this is normal V2. And this is the signal you get that reflects the action potentials of these cells. This is at the best orientation and direction. And again, you can plot the tuning curves. So the experiment involves putting the dread receptor into V1. And the idea here is that these layer 5 cells pick up the dread receptor, and they not only send it to terminals in Pulvinar, but these axons branch quite a bit, and there are many other targets that could potentially affect V2. So to make sure that we're just looking at this pathway here, we put the CNO directly into Pulvinar, which will affect only this synapse and not other synapses of this pathway. And when we do that, in many cases, we get a dramatic reduction in responses and visual responses because we're blocking this pathway. Here's another example. Some cells are not affected. To show the population activity, there's a surprisingly dramatic effect. Almost all cells, with some exceptions, have their responses dramatically reduced. This shows what's happened if we put an artificial CSF instead of the CNO as a control. There's some effect here, but it's much, much less than here. Statistically, a big difference between these two. And this just shows a couple of examples of tuning curves before and after. Now, this is a surprise to us because we didn't expect the transthalamic pathway to be so powerful. This tells us two things. This transthalamic pathway is necessary for the responses of most V2 cells. We don't know if it's sufficient. But we can say the reverse, and while the direct V1 to V2 pathway is still present, that pathway we would conclude is not sufficient for activation. Perhaps this isn't surprising because if you knock out the thalamic input to V1 from LGN, you won't get V1 responses. And it may be the case that the thalamic input to any cortical area is a lot more important than we generally think. But that's all speculation for now. Okay, I'm going to switch gears now and consider, yeah.
Sorry, I hate to ask this question. Did you do the CNO injection without dreads?
I'm sorry?
Did you do the CNO injection without dreads?
Yes, there are a whole series of, I should have emphasized, this is preliminary. The one we've done a couple of controls looking at because the injections are done in a later time period, we're looking at early versus late time period. We're looking at injections without dread. We're looking at, well, as I showed you, artificial CNS when we have dread. So there are a variety of controls we have to go through. And we're far from completing them. Okay, I'm going to switch gears and I'm going to talk about efference copies. And the idea is that when there's a motor command for any kind of behavior, there has to be a copy of that motor command sent in to an early part of the sensory processing stream. The idea of efference copies are very important. Let me just take a few minutes to explain them. Consider what happens when you move your eyes. And we move our eyes about 3 to 5 times a second. We make these fast saccades. Physically, when you move your eyes, the visual world stimulus on your retina moves in the opposite direction. But you don't normally experience the world moving around as you move your eyes. The world seems stable. And the reason for this is that the motor command to move your eyes, a copy of that command is sent early in the processing of visual information so that it basically eliminates that the effect of the eye movement in visual processing, so that by the time the next sensory stimulus comes along, that's taken into account. The idea of efference copies was appreciated as early as the middle of the 19th century by Helmholtz and others. It was first identified in frogs and fishes in 1950 experimentally. We believe it's important for any animal that has any kind of complicated behavior. And it's important for us to disambiguate changes in the environment that are caused by the environment itself versus those changes that are induced by our own movements. That's an important distinction to make. So the question I'm going to try to bring up without answers but some speculation is, what is the source of this efference copy and how do you make a precise copy? Well, one of the things that we think might be involved are branching axons. Branching axons are ubiquitous in the brain. They're found all over the place. And one thing we know about branching axons in mammals, warm-blooded animals, is that That's the most efficient and error-free way to get exactly the same message, the same pattern of action potentials down all the branches to the targets. It doesn't mean the responses postsynaptically will be the same because you would expect these synapses to be different. But if you want to copy a message to multiple targets, branching axons is a nice way to do it. And branching axons were first emphasized by Cajal. He did this over 100 years ago. This just shows one of his drawings of spinal afferents, primary afferents coming to the spinal cord. He emphasized very strongly the fact that every one of these axons branched, one branch going to the spinal gray matter, the other branch ascending towards the brain. This is a modern textbook view of that drawing. And we would call this branch going into the spinal gray as a message to the spinal gray, this message going up to the brain. We think of this then as a motor message because it'll have a fairly strong impact on motor neurons. Maybe not directly, in some cases it might be directly, but we think this would be strongly influence motor neurons, and we call that a motor message. We normally think of the branch going up to the brain as carrying a sensory message about the environment, a change in a joint angle or skin indentation. But because of the nature of the branching here, this would also be an exact copy of this motor message. And an exact copy of a motor message is a definition of an efference copy. Now, one point I want to emphasize is that the message going up to the brain is a single message. It's a single pattern of action potentials. It can be analyzed by some set of postsynaptic targets as a sensory message, and by others as an efference copy. So the idea that you have a single message that can be analyzed differently shouldn't be a problem. The message can be multiplexed in this way. So why do I say all this? Well, if we go back to our diagram, here are driving inputs that are innervating thalamus, and this information is related to cortex. We now know from anatomical studies from many labs that most, if not all, of these axons are branches. And that tells us that the message being relayed through thalamus is a copy of a message going elsewhere, and in many cases, the extrathalamic targets are motor centers. I'll give some examples of this. These are retinal axons taken from the cat. They have branches that innervate the lateral genicular nucleus of the cat, but you can follow the extrathalamic branch to the midbrain where it innervates the superior colliculus and pretectum. Every retinal branch that we looked at does this, every retinal axon. And these are centers that we can interpret as motor centers because the colliculus controls eye movements, the pretectum controls pupillary size, focusing, and so on. Here's an early example from Cajal, and there are many of these in the early literature. He's showing thalamic inputs from the deep cerebellar nuclei to the motor thalamus. And again, he emphasized that these are all branched, and the other branch goes to brainstem structures that we can identify as motor because they go to the red nucleus, which controls, it's a bulbospinal control nucleus for the rubrospinal pathway. This would be the reticulospinal pathway. And there are a number of these examples. Here's perhaps the best one. It's a relatively recent paper by Kita and Kita. We looked at layer 5 cells in motor cortex that are pyramidal tract, which means these axons actually enter the pyramidal, the spinal cord, and can presume to have some fairly strong motor effects, multiple branching all over the place. They showed a number of examples like this, all look the same. Here's the thalamic branches. So here's your transthalamic pathway. But this message is copied, a copy of messages to many other centers. This message is going to the spinal cord as a motor message, and there are other centers that we can identify as bulbospinal control centers. So these are all examples of branching axons that involve the transthalamic pathway that seem to be carrying a message that in some cases is a copy of a motor message. So to sort of sum up, the idea is that information comes into thalamus from the periphery through a first order relay, can be sent to a primary cortical area, and then we can go through transthalamic routes as much as we want up a cortical hierarchy. But there are direct connections as well. And I raised the questions about this. One insight as to what's different between these two pathways is these direct connections, with very rare exceptions, represent axons that have no subcortical branches. So this is information that stays entirely within cortex. Whereas these pathways, for the most part, have branches, which means this information is shared with other parts of the neuraxis. That's an anatomical fact. Many of them are motor centers, and we're trying to make sense of that. The idea that they're efferents copies, I'll remind you, is our speculation. You don't have to agree with that. But I am drawing attention to some anatomical facts that sort of beg for some experimental testing. Okay, so the final speculation now has to do with comparing our view to the conventional view. Conventionally, the idea is information comes into the thalamus from the periphery. It goes to sensory cortex, goes up a hierarchy through several stages of sensory motor processing, finally gets to a high level of motor planning, an executive area where a message is sent out eventually to control behavior, and somewhere at the end of this might be a single final efferents copy. Now there's several challenges we make to this. One is that I would argue that, and this is all hand waving, that this makes no sense from what we think we understand about evolution. Because evolution, we think, does not produce a new sensory receptor or sensory element without having a fairly immediate behavioral effect or motor effect, because such a process wouldn't survive evolution. So the idea that evolution would provide a circuit that took so much processing before you get any effect, any benefit from it, we don't think makes much sense. Again, that's pure hand waving. So the idea of a single entry and an exit point for cortical processing, we don't think is sound from a perspective of evolution. This also has no role for most of thalamus that we're calling higher order. The alternative view does give us fairly immediate potential motor effects as the information is processed through thalamus, through these branches. And as the information it has more computational power behind it, we update the motor command, and that update, that message, is sent through thalamus and up the hierarchies that continues. A couple things I want to emphasize is that these layer 5 outputs are found from every cortical area, including primary sensory areas. So the idea that we have sensory and motor cortex, we're suggesting is an oversimplification. Every cortical area has a motor output. The differences between what we call sensory and motor is more quantitative than qualitative. I'd also emphasize that cortex, for all of its beautiful computational power and circuitry, would be utterly useless except for the fact of these layer 5 outputs sort of represent the only way in which cortex can interact with other parts of the central nervous system and spinal cord to affect behavior. Without these outputs, there would be no effect on behavior. So it's important to recognize that, I think. Finally, just to remind you, we have these direct connections as well with lots of questions that arise from that. I want to emphasize that if you want to send an efferents copy between cortical areas, you don't have to pass it through thalamus. So the question remains, why we use a thalamic relay for whatever this information route is. Final point I want to make is that evolution is messy. One way to look at the evolution of efferents copies is that from an early vertebrate ancestor that operated mostly on spinal circuits, motor commands involved in these spinal circuits would have had to have efferents copies associated with them, and I've suggested one way of doing this. But as we developed, as evolution produced more flexible ways of controlling behavior through bulbospinal mechanisms, the rubospinal nucleus, tectospinal nucleus, and so on, each time a new motor circuit was evolved, efferents copies had to be evolved along with it, and the same ultimately applies to cortex. So if cortex is going to produce an output that creates a behavior, we're asserting that there should be an efferents copy associated with that. However that efferents copy is produced, we're suggesting one possible anatomical circuit for it, but again, that's just speculation. A problem that I don't think is recognized is that as a result of evolution, we have efferents copies at many levels, hierarchical levels that reflects evolution, that the brain has to sort out somehow. I don't have any suggestion for how it does it, but I want to suggest that this is a problem that needs to be addressed. So to summarize, what we're saying is that cortical-cortical communication appears to depend heavily on higher order thalamic relays such as the Pulvinar. Why does cortical-cortical communication seem to involve two parallel pathways direct and via thalamus? What's different about them? Why is one sent through thalamus? These are still open questions. How common is a parallel organization of direct and transthalamic pathways for cortical function? We have very few examples. We'd like to have many more to see how common this is. What is different about the information content in each pathway? Why is one processed through thalamus? And finally, what is the significance of the branching axons? Now, I want to emphasize for the last time that the branching axons do exist. They are an anatomical fact that is now pretty well established from many labs. Ephraim's copies ideas that I've thrown out are just our speculation. You can accept or reject that. We hope to have ways of testing these ideas, and if they get rejected, fine. But we do have to, I think, strive to get answers to why this anatomical pattern seems to apply to our thalamocortical processing. I'll leave it there. Thank you.
So I would just like to clarify something. In that one figure that you keep showing over and over again, the higher order, like the Pulvinar, for example, the different subdivisions that receive input from different areas, do they talk to each other or not? Because you never show this one arrow.
In thalamus,
Yeah.
there are no connections that have ever been identified between thalamic relay cells. So there's no, the only communication that's ever been suggested for thalamus is you can go from a relay cell in thalamus to the thalamic reticular nucleus to another relay cell. But there are no direct connections between thalamic relay cells. So
That's inhibitory, right?
that would be inhibitory, right?
How do you fit the direct pathway from cortex area 1 to 2 and into the driver modulator framework? You know, so you've said that thalamus to cortex is a driver input. What about and there's also modulatory inputs from thalamus to cortex 2. How do you fit that one in?
The modulatory inputs do exist. We haven't studied those in the higher order relays very much. The ones we've seen have been all driver. Cortical can be both. And we know in layer 4, for example, that you get a driver input through the transthalamic pathway, and the cortical can be either driver or modulator, depending on what layer of cortex they come from. That's what we've been finding. So there is convergence, and the convergence is complicated depending on the nature of the direct pathway and what layer it comes from. And what that means, we don't know. I mean, that's another open question. Yeah.
Can you make quantitative predictions what has to happen if you have the second pathway through thalamus concerning like delays, whether they are matched, the direct and the indirect, the type of codes that have to be used in order to make this match compatible?
No, I can't, but one thing I can tell you is that the latency, suggestions of latency of the two pathways doesn't lend itself to any simple answer. And the reason is, if you think of the visual system, the direct connections between V1 and V2 that are representing central vision are very short because of the topography. If they're representing an area 15 degrees out, they're very long. So the latency should be short for central vision and longer for peripheral vision. To go down and up is the same latency for either of those. And those are the fastest conducting axons in the brain, by the way. There's also a direct connection between V1 and MT, which is very long. So for example, we would say that the latency of the direct connection for central vision is much faster than the transthalamic one. The transthalamic one between V1 and MT is much faster than the direct one. So I don't see any obvious pattern there if our calculations are correct. Yeah.
So I have two questions. One is, in the experiment that you showed when you, the direction selectivity went away in V2 when you deactivated Pulvinar. Which layer of V2 were you recording from? Maybe it wasn't the slide.
In the whole animals,
Yeah.
I should have emphasized that. We can do the calcium imaging only down to the bottom of layer 3. So what you're seeing are layers 2 and 3. We are now using electrode arrays to see if deeper layers are doing the same thing. It's a great question. I forgot to mention this. So whether what we're seeing in the upper layers reflects what's going on in the lower layers is still an open question.
So the second question is, have somebody recorded a cell in Pulvinar that said, oh, this is a direction selective cell?
Oh, yeah, so, yeah. Pulvinar, people that have, there's a long history of people looking at receptive fields in Pulvinar. The complexity is all over the map. And part of the problem is that Pulvinar gets an input from 30-some visual areas, each of which has their own specializations for how they respond to visual stimuli. One conclusion you can make is the complexity you see in Pulvinar is consistent with the complexity you would expect from layer 5 inputs from all these areas. Trouble is you don't know what area is innervating a particular Pulvinar cell. So the simplest parsimonious explanation is that Pulvinar cells, like geniculate cells, are simply reflecting their layer 5 inputs for the receptive fields. Whether Pulvinar cells are putting together inputs from different cortical areas is an open question, and I think it's an important one.
So the general rules for the branching axons, I mean, you focus this on sort of sensory and motor areas. As you get up towards, say, prefrontal cortex and areas like nucleus reunions, PMD, and so on, do you see the same sort of thing?
You see the branching. This is an OR data. This is data. And there's a problem with branching axons. The database is pretty thin because it's very difficult to demonstrate branching axons. You can do it by double labeling, but if a negative result means you may have missed one of the areas. You can do it by anadromic activation. Same problem applies. So to me, the only good way to do this is actually labeling the entire the cell in the axon arbor with intracellular or juxtacellular labeling. There are very few examples of that because it's very difficult. What's striking to me is every example that I can find in the literature where that has been done, the axons branch, and it's from multiple cortical areas.